What I was asked to talk to you guys about today is to kind of illuminate the genetic basis for mental disorders. And there is a wide range of perspectives on what causes mental illness. And we're beyond the point of debating if it's nature or nurture. It's both. The question is, how much of it is nature? How much of it is nurture? What are the specific genes that put people at risk? And then we're not even really at the point yet of saying, well, what are the environmental factors? It's actually, easier to find genetic risk factors than it is environmental factors. Even though your own exposure to genetics is a science, you may think of it as very complex, it's actually more finite. The environment is so vast, so unmeasurable. We've just begun to scratch the surface. So I study genetics. So as mentioned, I'm at SUNY Upstate Medical University. I've been there for 11 years in October. I love it there. We do great research there. It's not Harvard. We don't do everything exceptionally well, but one of the things we do really well is neuroscience research, and especially psychiatric genetic research that I do. So I'll tell you a bit about my own research today, but also more importantly, the larger context in the field of what are we learning about the genetic risk factors for psychiatric disorders. So up front, I think it's important, you think about conflict of interest. When this person's telling you about anything, science or business, do they have a personal stake? I have a personal stake in my work because it's important to me. I also have a financial stake, and of course, science is a human endeavor. It does not get done in a vacuum. That's why there's way more research on autism now than there was 15 years ago. Research on every illness doesn't proceed systematically and equally. It's affected by how loud the voice is, and that's why NAMI is so important too. Is the National Institute of Mental Health going to get more or less share of the financial pie this year than the National Cancer Institute? Okay? Advocacy matters. Okay? So what I'd like to do at first is just acknowledge that none of what I'm going to show you relates to my own personal gain or well-being. I do get research grants from the National Institutes of Health as well as other organizations, and royalties from Oxford University Press for my book. And we've learned as a field that it's important to tell people up front who's paying for my livelihood, who's paying for the things I'm telling you about. Because if you remember, about 12 years ago, there was a doctor named Wakefield who published a study that suggested that the measles, mumps, and rubella vaccine caused autism. His financial conflicts of interest were not disclosed, but in fact, he had a huge financial stake in a company that was suing vaccine makers. Okay? So there's a conflict of interest between the science he's peddling and what he's making on the back end. And now, because of that one paper that was retracted, people aren't getting measles, mumps, and rubella vaccines. They're instead getting measles. Kids are getting sick and dying. Okay? So money can infiltrate everything. Science is a human endeavor. It's not some ivory tower activity that's disconnected from society. It's done by people, and every person has his or her own biases. So these are mine. I run what's called the Psych Gene Lab, the Psychiatric Genetic Epidemiology and Neurobiology Laboratory, which is a mouthful. So we abbreviate this as the Psych Gene Lab at SUNY Upstate. And who is the Psych Gene Lab? I've had great mentorship in my career. Ming Swong was someone who mentored me when I was at Harvard, along with Steve Rohn. Nick Shork's a guy at UCSD, and Frank Middleton's a faculty member who I work with at SUNY Upstate. A lot of these people here are my graduate students who've done a lot of the work in the lab. I remember what it was like to be a graduate student. It was a fun time. It was a hard time, but you got to do real science. Now, I run the lab, which means I write grant proposals. I try to get funding to pay everybody's salaries, and it's like, and they get to be in the lab and have fun. So these are the people doing the work and having the fun, and then I get to come and tell you guys about it. And then we've had sponsorship from a lot of foundations, including what used to be known as NARSAD is now the Brain and Behavior Research Foundation, has funded a lot of our work as well. And I'm very grateful for that. I couldn't do it without it. Research doesn't get done without funding. So how did I get back here? I went to Proctor High School in Utica, New York, came out here to Syracuse University, and I thought I wanted to be a marine biologist. I think we all thought we wanted to be marine biologists at one time, didn't we? And then I started taking biology classes and learned what it took to be a marine biologist, and I thought, this really sucks. You're not out there swimming with dolphins, and you have to learn hard science, and I wasn't ready for it. And I actually got a D in genetics at Syracuse. And here I am telling you today about it. So my sophomore year in particular at Syracuse University, there are a lot of difficult conversations and a lot of bad grades. And I found psychology as a second major, and that just clicked for me. That just resonated for me. So I did end up finishing. I got my bachelor's degree in biology. I persevered. But I also got it in psychology. And then for graduate school, I went to an experimental psychology program at Northeastern where I put the two together. In my third year in graduate school, I still didn't know what I wanted to commit my life to. I just knew I wanted to keep learning. I wanted to keep asking questions and doing research. And I took a long, hard look, and I decided that I wanted to try to find the ways in which genes and environments combine to make people sick. And not just make them sick, but to create mental illness. I thought, we're humans. What's more uniquely human than mental illness, okay? And what's more complicated? I figured by this point in my career, AIDS would be solved and cancer would be cured, and what's left is mental illness. Well, we're not there yet, but I still think at the end of it all, the brain will be shown to be much more complicated to understand than any of these other organ systems, and I want to dive into the really complex problems. So I did my postdoc at Harvard at Mass Mental Health Center. I did 3 years in San Diego, and then 11 years ago I came back to Syracuse because I saw a really nice opportunity at Upstate, and also my family's from the East Coast and wanted to be closer here. So the mission of the lab is that we'll develop and apply methods in pursuit of the determinants of mental health and mental illness. And the vision, what I want to accomplish in the lab, that we'll discover those determinants of mental health and illness, and then develop interventions founded on those discoveries. Now, interventions doesn't just mean new medications, but that's a part of it. We do need better targets. We do need better medications, okay? Every other field of medicine seems to have a much bigger stake in pharma than psychiatry does. The drugs work for maybe 50% of the people. 50% of patients on antidepressants don't respond. 50% of patients on an antipsychotic don't respond. Side effects are terrible. The problem is, we didn't go into the lab knowing what's wrong and then try to fix it. We started psychopharmacology by throwing drugs at people who were sick. Drugs off the shelf, see what works a little bit, and then tinker with that. The basic formulation for antipsychotic medications has not changed in 50 or 60 years, okay? So maybe it's new drugs, maybe it's early interventions in the environment, okay? We don't specify in terms of our vision, it's just we need to figure out why people get sick and what we can do about it at the earliest possible age. My first graduate student and I, he was more of a medical model guy, and I'm a psychologist, and he said, well, we should be focused on finding a cure. As sad as it is, I just don't believe there are cures to be found for mental disorders. I think it's about prevention. I think if we could keep people from suffering and having to go through that, then that's better serving the public good as well. But we keep pursuing those ideals. So our goals for today are to recognize the major genetic methods applied in the study of psychiatric disorders and understanding the genetic architecture. What are the genes and how do they interact to put people at risk for mental disorders? And you'll hear me talk a lot about risk or susceptibility, okay? We're not talking about genes that cause mental disorders. We're talking about genes that increase your risk. But there's other genes and there's other environmental factors that interact with that risk or that offset that risk, okay? These are not genetic disorders, but they have a genetic component to them. The basis for what I do is an extension to the field of genetic epidemiology. Epidemiology is the study of the distribution and determinants of disease in the population and with the transmission of disease in families. So there are two arms to genetic epidemiology broadly, population-based and family-based. We do both of these types of studies, and they both have advantages and disadvantages. So the questions of genetic epidemiology proceed fairly formulaically like this. Is the disease familial? Okay, does it run in families? If it doesn't run in families, Our job as genetic epidemiologists is done. Okay? There's no need to look for genes for the disorder. It doesn't run in families. If it does run in families, what are the relative contributions of genes and environments to the disease? Okay? Once we know the relative contribution, then we ask, what are the mechanisms? In what way are these genes transmitted? Okay? And then what are the specific mutations in our DNA that account for that genetic component, that heritable component? And finally, once we know the specific mutations, that's not enough for us to design those interventions I talked about. Knowing someone's at risk doesn't help us help them. We have to know how a specific mutation changes your biology so that we can intervene in that pathway with a medicine, with a dietary supplement, with some behavioral modification. So in a nutshell, this is what my work brain looks like. At the heart of it all is prevention. What's stopping us from preventing mental disorders? There's this big black boundary. We can't prevent a single case of mental illness right now. Why not? We don't know the risk factors well enough. Okay? We don't have any biomarkers. Okay? If you have concerns about mental illness in a loved one, you're all probably familiar with the process. You go see a specialist. There's a lot of uncertainty. There's a long observation. There may be diagnostic switching. Have we got it right? Have we not? Which medication? It's trial and error. We don't have biomarkers. We don't have an objective laboratory-based test like a biopsy that says, yes, you have schizophrenia. No, we don't have it, okay? Knowing risk factors and biomarkers does not allow you to prevent anything. It's knowing the mechanism, okay? How does having that risk factor change your biology? Or what's the mechanism by which that biomarker became disturbed? Okay, what's the biological mechanism? That's where you can intervene to prevent. And last part of what we do is focus on what's called phenotypes. So a genotype is your genetic constitution that you carry in your cells. The phenotype is the outward expression of that. And in the case of today's lecture, it may be things like schizophrenia. Schizophrenia may be a phenotype for research. Bipolar disorder. Any of your diagnostic categories in the DSM. These are what we call phenotypes. Why is the border around this circle fuzzy? It's because the borders around the phenotypes themselves are fuzzy. Okay? If you have followed the evolution of the Diagnostic and Statistical Manual since DSM-3 to now DSM-5, the boundaries change. The numbers of symptoms that you need to meet criteria for a disorder change. How can that be? How can we change the diagnostic criteria for a real illness? And so my question is the DSM actually defining something valid? And the answer is probably not. But it helps in a medical model where you have to be diagnosed with something in order to get treatment. You have to meet some criteria in order to start treatment, okay? With cancer, biopsy, cancerous, treat, okay? In mental illness, we don't have that clear dividing line. And if you look at the criteria by which a clinician has diagnosed someone, there are many different ways for you to meet that criteria, okay? So it's all very foggy, okay? The way you define those phenotypes determines how well you can find risk factors and biomarkers that are attached to those phenotypes. So it's like a narrowing of the arteries because we have ill-specified phenotypes. The better we can define real, valid entities in psychiatry, the better luck we'll have at defining risk factors and biomarkers. The problem is it's very circular. We do research on the phenotypes as they're currently defined, and then we find some things that are attached to those phenotypes, but how do we iterate? How do we revise it? Okay? So I would argue we probably need to take a more agnostic approach to who's ill and who's not, and get rid of the diagnostic labels when we're doing research. So, upon this, maybe you thought my brain was scattered before. Upon this, we layer all these different methods. When I'm organizing, what research are we going to do? Again, I'm a human being. I have to choose what are we going to focus on. We can't do everything. I'll pick some certain methods that I think we have facility with, that work well, that are cutting edge. I'll pick disorders that people are yelling and screaming about and getting attention for, like autism. In 2006, I wasn't doing autism research. I was only doing research on schizophrenia, bipolar disorder, and substance use disorders. Then Cameron was born, my oldest. I started to think, gosh, if you have a child who's diagnosed with autism, not only is that child's entire life impacted, the entire family, the siblings, it's a family disorder, right? So developmental disorders took on a new light for me. But also, there was money there for autism research. It turns out, coincidentally or not, probably not, a lot of famous people have a lot of kids with autism. And they throw money at it, say, let's fund autism research, let's find a cure. Well, okay, I have a skill set that I can apply to any mental disorder. I'm not gonna go study cancer just because there's money in cancer, but within the space of mental disorders, what should we be studying? Well, schizophrenia, because it's common, about 1% of the population, and it's very severe. Okay, but now autism is increasing in prevalence. There's money there. Oh, let's apply our skills there. Also have a personal stake in that. So we can't study every disorder, nor can any scientist. If a person has a personal stake, such as a family member affected, they may say, yes, this lab's going to focus on ADHD or PTSD or depression. But otherwise, we have to be nimble. We have to respond to what society tells us they want to know about. Okay, so again, your advocacy works. Okay. So now we're going to talk about the nuts and bolts. Here I've given you the background mindset, like where is my mind? And now I'm going to talk about the nuts and bolts of the genetics. Phenotypes in general, not just in psychiatry, can be broken down into two different categories. One are your Mendelian disorders. They're named after Gregor Mendel, the guy who did the experiments with the pea plants. He discovered, although he probably fudged his data, it's a very interesting story. Gregor Mendel was a monk. He kept meticulous records on the coats of peas, like was it smooth or wrinkled, the flower colors, etc. He discovered the laws of Mendelian randomization based on observing pea plants. People have gone back to his original records and said he probably tossed out some experiments that didn't conform to his expectations. Nevertheless, his laws are largely true that there are some things in life that you inherit in a one-to-one manner based on your genes, okay? And your blood type is one of these, okay? There's no environmental influence on what blood type you will get, okay? Eye color is another, okay? Nothing in psychiatry is really like this. However, there are a lot of Mendelian disorders that have a psychiatric component to them, okay? What we're talking about in psychiatry is mostly what we would call not simple or Mendelian disorders, but complex disorders, okay? Complex disorders are things for which you cannot find a Mendelian form of inheritance. In other words, it's not a dominant feature through a family pedigree. It's not a recessive feature. It's some members of the family have it, others don't, and it doesn't fit a pattern, okay? Or maybe there's only one person in an entire family that has it. It doesn't fit the simple genetic architecture. So sadly, for patients, and for all of us, most psychiatric disorders fall under the umbrella of complex disorders. How do we study complex disorders? There's two different techniques, two different genetic techniques. One is called behavioral genetic techniques. These are genetic methods that do not require a blood sample or a hair sample or a saliva sample or any DNA. These are genetic methods that can be done only by looking at families, talking to family members, and understanding the biological relationships between them. The second set of methods are the molecular genetic methods. These are the methods where, in fact, you've got a DNA sample from somebody, and now we're specifically gene hunting. We're looking for specific mutations, okay? But it all starts with behavioral genetics, and so shall we. So here's an example of one of the most basic methods in genetic epidemiology, a behavioral genetic method where you don't need DNA. It's called a family study. And a family study begins by identifying what's called a proband. That's the person who's identified here with the arrow. The proband is the person who has the phenotype we're interested in studying and who's brought the rest of the family to our attention. So if any of you guys have been involved in research, if you've been the proband or if a family member's been the proband in the study, that's what it means. It's the person that brought the family to our attention. Now in this case, let's say this is a study of depression, okay? In a pedigree drawing, the older generations are at top, the younger generations are on the bottom. Females are circles, men are squares, obviously we're square. And then if the circle or square is colored in, that means that person has the phenotype of interest, okay? So in this case, this proband has depression. Neither mom nor dad has depression. Maybe the maternal aunt may have dysthymia, some low-level depression. Doesn't meet criteria for major depression. But the maternal grandmother has depression, okay? Here's an example where the control or comparison subject proband who brought his or her family in does not have depression, and maybe her mom has dysthymia, but nobody else in the family shows any semblance of depression, okay? If a disorder or a phenotype is genetic, you will see more families, more affected individuals among family members of someone who has the disorder than you will among family members of someone without the disorder. This is never black or white, and that's why I put this hashing here. And you may have cases of depression in a family, but the proband doesn't have depression. That happens. It's an average over many, many families. And on average, what we would see is that the odds of someone having the same disorder as the proband are much higher if they're in a family that already has the disorder. Okay? Now let's look at this another way. This is for schizophrenia. The percent risk for schizophrenia in the general population for any person who doesn't have a family member affected with schizophrenia is less than 1%. It's about 0.8%. If you have an aunt, uncle, niece, nephew, or grandchild, or half sibling with schizophrenia, your risk is about double that. It's about 2% to 4%. If your parent has schizophrenia, if your sibling has schizophrenia, or one of your children has schizophrenia, or you're the dizygotic or fraternal twin of someone with schizophrenia, your risk is anywhere from 4 to 10 times the population. If you're a child of two parents who have schizophrenia, or if you're the monozygotic or identical twin of someone with schizophrenia, your risk is about 50%, okay? So 50 times the population base rate. This graph on the right shows how many genes you share in common with these people. In other words, is it a first-degree relative, a second-degree relative, okay? And so we see that as you increase the genetic similarity to one of your family members, you're a closer family member, and if they're affected with the disorder, your chances also go up, okay? Now, this is a table that I put together many, many years ago. I've tried to update as much as I can with the results from family studies of as many psychiatric disorders as I could get my hands on. And what you see here, this lambda value, is the relative risk. What it means is, if I'm a male, if my brother has autism, what's my risk of autism? For a female, if my sister has depression, what's my risk of depression? So lambda relative risk means same-sex sibling risk, okay? And what you see is that at the top of the list is autism with a lambda of 22. That means if the base rate of autism is 1%, which is actually a little higher than 1%, if the base rate in the population is 1%, if I have a sibling, a brother with autism, my risk is 22% or 22 times higher. Okay? And for schizophrenia, for example, the relative risk is 12. So the population risk is 0.8%. If my brother has schizophrenia, my risk is about 10%, or 12 times higher than the population. Does that make sense? Okay. Relative risks for mental disorders span a really wide range, from 10s to 20s, all the way down to 2s to 4s, depending on the disorder. Okay? So there's a wide range here. But there's none for which the relative risk is 1. If the relative risk is 1, that means you're at no more or no less risk than someone in the population. For every single psychiatric disorder I've looked at, the relative risk is always higher than 1. At a minimum, it doubles your risk. At a max, it may give you 22 times the risk. Now, family studies have limitations, okay? Not everything that runs in the family does so because of genes, okay? You think about your own families. Your parents spoke English, so you speak English. Your parents may have been of a certain political orientation, so you may be, or you may rebel against that. But the point is, a baby is born with the inherent capacity to speak any language. or have any political affiliation or religious affiliation. Why doesn't a baby born to English-speaking parents speak French? It's learned, okay? Things run in families for other reasons besides genes. Learning, viral transmission, okay? Tuberculosis. If you look at family risks of tuberculosis, they look just like that. And it's because the closer you are to someone, the more likely you are to live with them and to share this disease, okay? So things don't always just increase in families because of genetics. But they must show some evidence of running in a family in order for us to suspect that genes play some part, okay? Now, we've shown that for every psychiatric disorder that we've looked at, they do aggregate in families. Do they aggregate in families because of viruses, because of learning, or because of genes? We can't tell from a family study, but twin studies and adoption studies can help us. Adoption studies, we don't do too many of anymore because of privacy rights. It's very hard very interesting design, but we'll mostly talk about twin studies. Twins give us a really beautiful, elegant, natural experiment, okay? Twins come in two types. Monozygotic twins come from one single fertilization event. So in a pedigree, we'd show it as one single fertilization event that then splits in two. Okay, monozygotic meaning one zygote, identical twins. This twin shares all of their DNA in common with this twin. Okay? They also occupy the same uterus. They live in the same home. We study twins who are only raised together. Sometimes twins are raised apart. We don't study them. Dizygotic twins, on the other hand, are two separate but contemporaneous fertilization events. So we show them branching off right at the time of conception. Two eggs, two different sperm. Therefore, these twins share, on average, 50% of their genetic material. No more identical than any other brother and sister, or brother and brother, or sister and sister, okay? No more identical than any other sibling pair. But they do share the same uterus, and they do live in the same household. So the only thing that's really different about monozygotic twins and dizygotic twins is 100% genetically identical, 50% genetically identical. Okay? By studying pairs of twins with mental disorders, we can figure out how often, if one twin has a disorder, does the other twin have a disorder. And if genes are important for that disorder, then more monozygotic twins who share more genes in common are likely to be concordant or have the same phenotype. More so than dizygotic twins who only share 50% of their genes. Okay? Is that clear? Okay. We do the same sort of inspection, and what we see is not surprising, is that those disorders that ran the most strongly in families also have the strongest concordance among monozygotic twins. So for schizophrenia, I showed you already, if your monozygotic twin has schizophrenia, you're about 50% likely to have the disorder yourself, okay? It's not absolute, and that fact alone indicates that these are not genetic disorders, okay? There's room for the environment to operate. But those disorders like autism and schizophrenia and bipolar disorder and obsessive compulsive disorder that had the strongest family contribution also have the strongest genetic contribution. In other words, they didn't run in families because of viral transmission or learning. They ran in families because of genes. Okay? And there's way more twin studies than there are family studies, by the way, so lots more disorders on the table. For every disorder I've looked at, except for dysthymia, which is relatively rare, actually, in research, every other disorder has some heritability that is greater than 0. What that means is that heritability can range from 0% for a trait that's only influenced by the environment to 100% for a trait that's entirely genetic, okay? Our top disorder was autism at about 92, 93%, okay? That means that 93% or 90% of the risk for autism is genetic, okay? Now down at the bottom, most of those disorders have about 20%. That means most of the risk for those disorders, things like enuresis, social phobia, and a lot of personality disorders, is environmental, not genetic. So there's a broad range, okay? Now it's a complicated fact, but you can't take heritability estimates at their face and say, that's how much of the disorder is genetic. Things also can show increased heritability or decreased heritability based on the environment. For example, PKU, phenylketonuria, is a recessive disorder caused by a single gene. It's a Mendelian disorder. However, it's only expressed when you're exposed to phenylalanine. That's why if you have a Diet Coke and you look on it, it says phenylketonurics contains phenylalanine. Why does it say that? It's to warn people that this contains phenylalanine. Don't take this if you have PKU recessive mutations. Okay, children born with PKU about whom they don't know their genotype and are exposed to phenylalanine will have a severe mental retardation, but only when they're exposed to phenylalanine. So even though it's a genetic disorder caused by one gene, it won't express as mental retardation unless the environmental cofactor is there, okay? So if everybody's exposed to phenylalanine, the heritability of PKU is 1. It's 100%, but not everybody's exposed to it. So heritability is a little bit wishy-washy too. These numbers are the upper limit on how much of the variability in these disorders is genetic, but it could well be lower. This is the absolute upper limit of how much of the variability is genetic. The math is a little complicated on it, and we shouldn't get into it today. Okay, so twin and adoption studies tell us that these disorders run in families, at least in part because of genes. Environmental factors are also important. There is no mental disorder for which the heritability is 100%. Okay, remember autism at 90-something%. What's contributing the rest of that risk up to 100% is the environment. Okay? There's a lot of reasons that heritability is probably lower than what we've estimated from twin studies. For example, monozygotic twins are often much more similar to each other, not just because they share more genes in common, but because of their temperament, right? Or the fact that their parents may know that they're identical twins versus dizygotic twins and treat them more similarly and so on. So their environments are also more similar. Okay, these disorders run in families. They run in families in part because of genes. Are they Mendelian disorders? Are they caused by a single dominant gene? If so, every time we have one of the parents who has the disorder, we'd expect half of the offspring to have the disorder. That's Mendel's law. Is it caused by a single recessive gene? In which case, we'd only see affected children if both parents had the affected disorder or if they're both carriers for the disorder. Okay? We don't see that in psychiatry. Almost never. Okay? This is what we see in psychiatry. We see all the way back to the great-grandparental generation, nobody was affected with the disorder. Then in the next generation, there's a partially affected individual and there's an affected individual. And then the children of that affected mom may have varying degrees of being affected or none at all. Okay, it's incomplete penetrance. Okay, this is a complex, what's called polygenic effect. Poly meaning many genes. Okay, so it runs in the family, but not in any simple way. All of that is to say, they run in families, these disorders. Genes are somewhat responsible for that, but not just one gene per disorder, many genes per disorders. So we should be hunting genes, okay? And we made this decision about 25 years ago to start hunting genes for mental disorders. Now we're talking about getting DNA samples. We know these run in families and so on. Now let's go dig into people's genomes and find what are the mutations that are responsible. If you look at all those pedigree types, almost every one of them is what we would call polygenic. That means they don't transmit in a simple manner. There are some cases of some mental disorders that seem to transmit more in a simple genetic manner. Some families show a strong family pattern of obsessive compulsive disorder. Some families show a strong family pattern of major depression. I've even seen this in bipolar disorder. We've seen one family at SUNY upstate where there are 8 individuals with bipolar disorder in one family across 3 generations. So there are some families that are the exception to every rule. But by and large, every family affected by one of these psychiatric disorders shows a complex inheritance pattern, not a straightforward Mendelian inheritance pattern. Okay, same is true for most of them that have been looked at. Okay, so as I mentioned, the search for risk genes is warranted. We should be going after them. So there's one technical slide after this this is the extent of the deep science I want to get through. Okay, if you were like me, biology aversive, and never learned this, that's okay. I think if you learn one thing here today, an understanding of how we drill into the genome, how we look at it, and what these terms mean, okay? So in all of our cells, we have this nucleus, which is the center of the cell where our chromosomes are, which we've inherited from our parents, okay? And each of those chromosomes, you may have seen a representation of your chromosomes, either a picture or a cartoon like this. What is a chromosome? Well, we have 23 pairs of them. 22 pairs of them determine the proteins that we make that make up our skin and our enzymes and all these proteins. One pair determines what sex we are. You either have an XX in your female or XY in your male, right? Well, what is a chromosome? Okay, a chromosome is simply a very tightly wound strand of DNA. Okay, if you stretch the DNA out, it'll stretch here to the moon. But we can fit that in each of our individual cells because it's wrapped very tightly into a chromosome. Okay, so you see that unwrapping here, right? And then when you look at the DNA more closely, what you see is this very elegant mechanism. God built this, nature put it in, whatever you want to. I think it's beautiful and elegant, no matter who made it, that we have a built-in mechanism for replication, copying, okay? We can make copies of our DNA and then pass those on to our children, okay? When our DNA separates, we make transcripts of one or the other strand of that DNA. DNA is a double-stranded molecule, the double helix, okay? This is the double helix, okay? That unwinds from each other, and then we make a copy of one or the other strand, and then that transcript is then made into the proteins that we use as the building blocks of all of our cells. They come back together in a very specific manner. They replicate in a very specific manner. If on your strand at a particular position you have a guanine, on the other strand you will always have a cytosine, okay? If you have on this strand a cytosine, you will always on the other strand have a guanine. And the same pairing applies for adenine and thymine, okay? This is a rule book for life, okay? And thus when these DNA strands get separated from each other, they know how to replicate themselves. When I talk about specific mutations that are associated with disease, this is what I'm talking about. Where most of us may have a guanine nucleotide, people with a mental illness may have a thymine nucleotide. Okay? It's not that the pairing gets disrupted, both strands get swapped out, just compared to what the typical genome is. Okay? So how do we go about looking for associations between DNA mutations at a single nucleotide? There are 3 billion pairs of them, so 6 billion of these color bars in our genomes, okay? 3 billion pairs. How do we go about saying, aha, having an adenine at this position increases your risk for depression, whereas having a guanine at that position is the baseline, okay? How do we go about that? Well, we have to identify places in the genome where we all differ from each other. At most spots in the genome, we are not different from each other, okay? But there are about 10 to 11 million places where we may differ from each other. 10 to 11 million sounds like a lot, but that's out of 3 billion positions, okay? So we have a fair amount of variability, okay? We identify these places in the genome, and then we genotype these. That's done in a laboratory by basically knowing specific chemistry that reacts to those different color-coded bars, okay? So that's how I could take a blood sample from me and from you and from you and from you, perform this chemistry, and I'll know, okay, I have an orange bar, you have a green bar, you have a red bar, you have orange, green, red, and so on, okay? And then we ascertain families or individuals who have these disorders and say, are the people who have the disorder in any way representing more of one type of nucleotide compared to a matched sample of people who don't have that disorder? If so, then we can make an assumption that, well, possessing that particular nucleotide increased their likelihood of having a disorder versus not having a disorder, okay? What that looks like is like this. On the left, we have 4 cases. These are people with a particular disorder we want to study, and we may genotype them at one particular locus. You have 1 pair of chromosomes from your mom, 1 from your dad. So each person is going to have 2 markers, okay? This person has 2 reds, 1 red from mom, 1 red from dad. This person is red and blue, this person is blue and red, and this person is red and blue. Then we look at a matched sample of people who are alike these individuals in every way, except they don't have the disorder we're interested in studying. They're the same age, they're the same ancestry, everything, but they don't have the disorder. And they may also have 2 reds, they may also have red and blue genotypes, they may have blue genotypes. We would do this in thousands of individuals, and then we would tally it up. And if you see some disparity, in other words, if the number of red to blue alleles is different in our cases than it is in our control subjects, then again, we can make some assumption that having that red allele increases your risk. Now, having that red allele, or genotype, does not cause this disorder. If it did, then this person here would have the disorder, and this person here would have the disorder. It's just probability, okay? Having it increases your chances, but it's not absolute, okay? There is no gene that is necessary or sufficient to cause a mental illness. There is no single gene, okay? So a lot of genes have actually been found to have mutations that are associated with these mental disorders. Many, many genes have been associated with them. Some of them have not been studied at the genetic level. Why is that? Well, again, this is a human endeavor. We as a society have decided that schizophrenia is more important to study than narcissistic personality disorder. Although borderline personality disorder, I mean, you could argue this should be a high research priority. Nobody's really studying it. But nevertheless, the disorders that we as a society say are really important to study get the most attention, the most research dollars, and then we find the most about them, okay? Now, what this shows here is we have to study thousands and thousands of people before we find something reliable. And so when we were studying 1,000 schizophrenia cases and 1,000 control subjects, we didn't find anything. Once we got above 5,000, and especially once we got to 10,000, which this is logarithmic, so this is 10,000. Now once we have 10,000 cases and 10,000 control subjects, we can finally find genes that are reliably associated with an increased risk of schizophrenia, okay? And some other psychiatric disorders are starting to get to those same numbers of patients. But schizophrenia is the leader, okay? Actually, depression is highest now. I debate whether or not to talk too much about this, but anything above this red line, these are your chromosomes from largest, although smallest numbered. Chromosome 1 is the biggest chromosome. Chromosome 22 is the smallest chromosome. And these are all the different genes on all your chromosomes. And on the y-axis here, the up and down axis, is the degree of evidence or reliability that those genes are associated with an increased risk for schizophrenia, okay? There is not just one gene. There is 150 or so already, or 200 now, genes that have been found to increase your risk. This one here, though, by the way, this is on chromosome 6. This is the major histocompatibility complex. This contains human leukocyte antigen genes. In other words, these are genes that regulate your innate immunity, okay? And they have the strongest evidence for risk for schizophrenia. This is mind-blowing for the field, okay? We would have expected, well, you know, the genes that are going to be the strongest risk factors are dopamine receptor, because every drug that treats schizophrenia does so by blocking the dopamine receptor. Well, the D2 dopamine receptor is about here. This is the gene that encodes the D2 dopamine receptor. So the gene that encodes the D2 dopamine receptor certainly is an above-the-line, reliable risk factor for schizophrenia, but it's not the one that's the most strongly associated with your risk. And in fact, what we see time and time again, not just for schizophrenia, but for all mental disorders that are at this level of molecular analysis, is that there is no escaping the fact that immune and inflammatory genes increase your risk for mental illness. This is mind-blowing, okay? Because when you think about the drugs, antidepressants, the major class of antidepressants block one protein. They block the serotonin transporter. Okay, they're SSRIs, selective serotonin reuptake inhibitors. If you look at a plot like this for depression, you will not see the serotonin transporter anywhere there. Okay, schizophrenia, every drug that works to diminish symptoms blocks the D2 dopamine receptor. Well, it's there, but it's nowhere near the top. Okay, autism. Okay, we don't even have good drugs for autism. ADHD, what drugs work for ADHD? They're stimulants. They do the same thing for the dopamine transporter that antidepressants do for the serotonin transporter. If you look at a plot like this for ADHD, the dopamine transporter is not there. Okay, so we labored for 50 or 60 years with neurobiological models of these mental illnesses that were carved from the ground up based on anecdotal psychopharmacology. In other words, what drug works a little bit, that's the basis for our model, and let's design every experiment to focus in on that. When really, if you take the blinders off and let the genome give you the answers, it says, look at the immune system first. There is one study that I've been involved in called the North American Prodromal Longitudinal Study, or NAPLES. This study studies kids in the transitional age range, 18 to 21, who are starting to show signs of risk for schizophrenia. Social isolation, magical ideation, they're having thoughts that don't jibe with reality, and has followed them longitudinally to see what are the things that predict who's going to go on to have schizophrenia versus who's going to go on and develop typically. Within that study, they've now started to try anti-inflammatories. They've also started to try fish oils and some nutraceuticals to see, can we, if 40% of people convert to schizophrenia over 2 years, can we get that number to 20% or something like that? And there's some indication that it works, but not that well. So I think another point to this is that even though this massive tower is occupied by immune and inflammatory genes, even though that massive tower is the immune system, it's still not the only thing going on. It's just one component of it. So we need probably a much more complex medicinal chemistry. In other words, we need to build drugs that don't do one thing, they do 20 things. When we talk about a particular disorder, it's best not to conceptualize it as one, but as many. And the subtypes of that disorder, from a biological standpoint, may overlap like those in the middle. But there may also be some out at the edges that nobody else shares, that are unique, that are private. Maybe they converge at the level of biology, but at the genetic level, everybody's got their own constellation, right? So we need to change the way we do research. So the DSM guides the research, but the DSM is subjective. It's based on phenomenological descriptions of behavior instead of biomarkers. So that's why we have to continue with the genetic and the biological. So I'll take a couple last questions and then I'll push on. I don't know if that generates to country level data, but there certainly is good data that people who live in cities are at an elevated risk for schizophrenia, for example, compared to people who live in the country. But I don't know if that generalized like to the US with our work week and that lack of vacation versus other socialized Northern European countries. I'm not sure. The Northern European countries have things working against them too, like less sunlight and things like that too. And actually a concentration historically of some population isolates with a very high risk of schizophrenia. So those things balance out. But cities versus rural distinctions are well known. The question is it stress? Is it smog? Is it, do people with mental illness gravitate toward the city? We don't know. So it's hard to disentangle. Yes. Alzheimer's, is a tough one. It's on the borderline between neurology and psychiatry. Alzheimer's has very strong forms that are known to be attributed to a single gene. Unlike most of these psychiatric disorders, there are forms of Alzheimer's Alzheimer's that are caused by a mutation in one gene. There's presenilin 1 and presenilin 2. These cause Alzheimer's in the early 40s to 50s. Very early onset, very serious forms of Alzheimer's. And then there's most cases of Alzheimer's, which occur in the late 70s, 80s, that have ApoE as a strong risk factor and a lot of common complex genes like I've showed for these other disorders. But Alzheimer's has a known brain pathology. And so neurologists can kind of wrest that away from psychiatry. Like there's a thinking in medicine that every disorder is neurological if you look hard enough. The distinction is psychiatric disorders don't have a known substrate that you can point to, like plaques and tangles in the brain's tau pathology. Alzheimer's does, so it's more considered a neurological disorder. But it does have a complex polygenic contribution to it. It also has single genes that are causative for Alzheimer's. Yes. So there's two types of ways to do a family study. One of which is through the family diagnostic method, which means the proband comes in, the proband has a disorder, and we'll ask the proband, okay, what about mom? What about dad? What about your aunts, uncles, and grandparents? Depending on what disorder we're studying, the proband, him or herself, may not have a very good sense of who else in the family was affected with the disorder. They may diminish the number, they may exaggerate the number, they may not recollect the number, they may not be even coherent enough to answer those types of questions. So oftentimes we'll go to a reporter. Let's say if a child is affected with a disorder, disorganized schizophrenia, not going to be a good reporter on family history. We'll go to the mom or dad or somebody who's a close relative who knows the proband and knows the family history, and we'll ask. If somebody kept their illness very well hidden, we won't know, okay? And so the family history method will underestimate the level of symptoms in the family. And thus, lambdas that I showed you will be too low. They should actually be higher. There's another method that's rarely used because it's much more laborious and expensive, which is the family diagnostic method, which is, here's the proband's sick. Tell me not about your mom and dad. Tell me how to get in touch with your mom and dad. I'll go talk to mom. I'll go talk to dad. I'll diagnose them. Okay, where's the grandparents and so on. So in that type of study, somebody would have to be very good at hiding their symptoms and be willfully anti-research to kind of hide that. So, but family diagnostic method gives more accurate estimates, but is more expensive, and thus it's not done as much. Yes. There have been some beautiful studies done looking at home videos of people who went on to have schizophrenia decades later. And if you look at if you have blind research associates and research psychiatrists look at the videotapes of kids who went on to have a typical course versus kids who went on to have schizophrenia, they can tell. They can tell just looking at them, not looking at any diagnostic criteria, just looking at the presentation of a child in a home video in terms of their neurological soft signs like coordination and things like that. Those signs were there. We just didn't know what to look for. We don't diagnose them at that age. But the signs are there, and in fact, I would argue, and I think there's enough data to suggest this, that except for late life neurodegenerative disorders like Alzheimer's, cognitive impairment, etc., for things like schizophrenia, bipolar disorder, certainly for autism, ADHD, the architecture for these disorders is in place in utero. Before birth, the brain is being built in a way, according to a map that doesn't have the archetype of normal brain development. It's already being constructed in a way that's going to put someone at risk. And so, of course, along the way, there's going to be perceptible signs, but they're much more mild. That's not psychopathology. It's just lack of motor coordination, neurocognitive impairments, and so on. When the person reaches, for schizophrenia, bipolar disorder, late teens, typically, stresses are building, interpersonal stresses, finding a partner, going off to college, getting a job. The stresses are building. Your brain naturally is starting to prune off some of those connections that it made during early childhood. Our brain makes way more connections than we'll ever need. Redundancy to protect itself. If you have one neuron doing a job and that neuron's damaged, the job doesn't get done. But if you have 100 and one of them gets damaged, you can still do the job. Our brains do that. They make 100. And then we say, okay, we've got the job on order. We don't need 100 nerves to do this anymore. Let's start to get rid of some of them, okay? So the thought is that during that buildup where you have excess cognitive reserve, let's call it, neuronal reserve, there's no problem, okay? But once the normal brain pruning starts to occur, then coupled with the stress, that's when the symptoms start. Let me tell you a little anecdote. I used to be a guy who was just intrigued by scientific questions. And I went to graduate school and to do my postdoc in psychiatric genetics because of the challenge of studying brain disorders. This was not personal for me. My family does not have a strong history of mental disorders. The founder, so I won an award from the Bayer Foundation and from NARSAD, and there was a write-up in the Post-Standard up in Syracuse about this. And the founder of NAMI Syracuse, her name is Betty Pringle. Do any of you guys know Betty? Betty Pringle, she's the founder of NAMI Syracuse, wrote me a letter. And she said her son, Stephen, same name, has schizophrenia. He's had it for 35 years. She's watched, all these problems with him. And for the first time in my life, it got personal. And so I met with Betty, and she told me about NAMI, and I got involved with NAMI, and it changed my perspective on everything. And so finally, I'm like looking at the people behind the disorders I'm studying. These aren't just problems. There's people who have a stake in this, and you guys especially. So that's why I joined the board of NAMI. And one of the things I did with NAMI is breaking the silence. I get out in the schools, and I teach kids about mental illness, that these are biologically based brain disorders. This is the language to use, why stigma is so harmful, what to do to be more inclusive, to decrease social distance, to increase knowledge, to improve attitudes about mental illness. In the work that I've done on PTSD, including on Marines who were deployed to Iraq and Afghanistan, what I found as the number one distinction at baseline, before these guys were deployed to Iraq and Afghanistan, this was back when I was in California and they were training at 29 Palms in the desert, we would go take a blood sample from them. And then they would go off to Iraq and Afghanistan, are exposed to horrific trauma. They'd come back, 20 to 30% would develop PTSD. We have the blood samples from when they were in training. And we say, what's different in the blood samples from those who went on to have PTSD versus those who did not? And it's heightened immune and inflammatory gene expression at baseline can serve as a predictor of who's at the highest risk. If you want to ask me to theorize, I think at the heart of all brain disorder is an inflammatory and immune component. And then things like dopamine dysfunction or serotonin dysfunction or whatever, those probably push you more towards schizophrenia or ADHD or autism or depression. But the core risk for a brain disorder, including Alzheimer's and late life neurology, the core risk for a brain disorder is probably a faulty genetic architecture for immune and inflammatory genes. That's what if I had to guess. Another big component after immune signaling is cell adhesion. These are proteins that regulate how well neurons and other cells stick to each other, find each other and stick to each other. It's like the basic architecture of the brain. That's why I say, we won't find a cure for these disorders. They're probably caused by genes that fundamentally change the way neurons are built and hooked together. And neurons are non-mitotic cells. They do not divide. Like you constantly are making new blood cells. You're not constantly making neurons. And so if you stack your neurons, it's like building a building. If you put the bricks at the foundation together in a different way, and then you build a building on top of it, you can't go change the foundation later, okay? Or it's very difficult to do so. So that's probably the best analogy for